this video, we're going to focus on the cell cycle, which is a sequence of events of a dividing cell. It lasts from the beginning of one cell division to the beginning of the next cell division. The frequency of cell division varies by the cell type. Embryonic cells, for example, can divide about every 20 minutes, whereas if you have skin cells, these can divide every 12 to 24 hours, and liver cells divide once every year or two, but can increase the rate of cell division if the liver is damaged. Most mature nerve cells and skeletal and cardiac muscle cells don't divide after maturity. They are permanently what we call G0 or G0. Let's go into a little bit more detail about G0. Well, G0 or G0 is the normal functioning state of a cell that's not preparing to divide. It's doing the normal things that a cell does. Some cells enter G0 temporarily, while other cells never leave or divide again. For example, most mature nerve cells that I just mentioned, skeletal and cardiac muscle cells, they don't divide after maturity. And when you're looking at the cell cycle, this arrow coming out to the side is showing cell cycle arrest, or G0. It's not that the cell isn't functioning, it's just that the cell is not getting ready to divide. It's just doing its normal activities. Well, the cell cycle consists of two distinct phases. We have interphase, which is what you see here in purple, and we have the mitotic phase, which is in brown. We're going to go through each of these phases in detail. So we look at interphase. This is the time in which the cell prepares for cell division and consists of three subphases, which you see right here. We have G1, S, and G2, and then this triangular looking structure is the mitotic phase. Well, the G's just stand for gap. This is gap 1 and gap 2, and it's the gaps between the S phase and the mitotic phase. The G1 phase, or gap 1 phase, the cell is preparing for replication. And in doing so, the cell is going to produce all the enzymes required for DNA replication. Because remember, you got to duplicate your DNA if you're going to divide a cell into two cells. They each have to have a copy of the DNA. During this time, the cell is going to duplicate most of its organelles. Now, some organelle synthesis, such as the centrosomes, which you see here, they can take up to G2 to complete, but they're going to start in G1. So you want to make sure you note that. G1 is about cell organelle replication. Then the cell is going to enter the S phase, and S stands for synthesis. And what happens in the S phase is that the DNA is going to be replicated. So during the S phase, the double helix is going to separate, and then each of these sides is going to become a template for building the other side. When you're finished copying this, you're going to wind up with a chromosome that has one sister chromatid here and the other sister chromatid here, and they're joined by a centromere. This sister chromatid here is this part, is this right here. And this sister chromatid over here is this part right here. So they're exact copies of each other, and they're held together at a structure called the centromere until it's time for the sister chromatids to separate, which is going to happen during mitosis. Now the next phase is the G2 phase, or gap 2 phase. And this is devoted to last-minute protein synthesis as a cell gets ready to divide. It's the final check to make sure everything is replicated the way it should have been. The organelles are replicated. You have your enzymes ready to go for mitosis. That's what the G2 phase is for. Let's go ahead and test our knowledge on the cell cycle part 1. You want to select the term that makes a sentence correct. Go ahead and pause the video. Here are the answers. Pause your video, select the term that makes a sentence correct. Here are the answers. Go ahead and pause the video, select the term that makes a sentence correct. Here are the answers. We've gone through interphase, which is all of this here in purple. The next area we're going to focus on is the mitotic phase. And the mitotic phase is all about moving the chromosomes to where they need to go and then divide the cell. So let's take a closer look at the mitotic phase. The mitotic phase consists of mitosis, which is nuclear division, and cytokinesis, which is cytoplasmic division, and results in the formation of two identical cells. Looking at mitosis, this is division of the nucleus. So when we see the nucleus here, we've copied the DNA. We have our sister chromatids. Mitosis is all about arranging these sister chromatids so that they're at the center of the cell here, And then we separate the sister chromatids, and then each of those sister chromatids goes to opposite poles, and then you're going to divide the cell, which we call cytokinesis, which is division of the cytoplasm. The end result, you're going to get two cells with the exact same chromosome number and genetic information. It's important to note at this point that you replicated your chromosomes during the S phase of interphase. So now you have 46 chromosomes, but they're replicated and they're held by the centromere. As long as they're held together by the centromere, it's considered to be one chromosome that consists of sister chromatids. When you go through mitosis, those sister chromatids are going to separate. And at this point right here, when we're looking at a human cell and these sister chromatids separate, you go from 46 chromosomes here to 92 chromosomes. You're going to have 46 chromosomes on this side and 46 chromosomes on this side, which means the next phase is going to be cytokinesis. You're going to divide this cell into two cells, each with 46 chromosomes. So let's take a look at the mitotic phase in a little bit more detail. So we're going to look at the details of mitosis using this animation. Mitosis consists of four phases. We have prophase, metaphase, anaphase, and telophase. And I'm going to go through and show you in this animation each of these phases. But before we can get to this stage of mitosis, we had to go through interphase. And in interphase, remember, we had the G1, S, and G2 phase. So here we have a cell. This is the outer membrane of the cell, or the plasma membrane. The organelle that's shown here is a centrosome with its two barrel-looking structures called the centrioles. These are going to produce microtubules. This structure right here is the nuclear membrane or nuclear envelope, and inside here is the chromatin. That's the DNA. This cell is getting ready to divide. And so during this time, during this interphase, you replicate your DNA during that S phase. You're also replicating your organelles. Remember that G1 phase, you replicate your organelles? Well, let's watch what happens when you replicate the organelle. And the only organelle we're showing here is the centrosome. There we go. We have replicated the organelle. So now we have two centrosomes. Well, the S phase is when your DNA is replicating, but you can't see the chromosomes yet because they're still in the form of chromatin. That's loosely wound around this histone protein so it can open up and copy the DNA. You go through the S phase, you go through the G2 phase, and then you enter prophase. Let's see what happens during prophase. The nuclear membrane begins to disappear. The centrosomes are going to migrate to opposite poles. Now notice what they're producing as they're migrating to opposite poles. They're producing spindle fibers. These spindle fibers, which are made of microtubules, and you can call them spindle fibers, you can call them microtubules, are going to attach to the centromere of the chromosomes. And the nuclear membrane has to disappear so those spindle fibers can get in and attach to the centromere. Let's look a little bit further into prophase. Take a look at this. The nuclear membrane's almost completely disintegrated. We can now see the chromosomes. We've gone from chromatin to tightly wound around the histone proteins called chromosomes, joined at this circular structure here called a centromere. So we can see them now underneath the light microscope. The centrosomes are migrating to opposite poles, and they have the spindle fibers coming out, which are going to attach to the centromere. The next phase after prophase is metaphase. In metaphase, the sister chromatids are moved using these spindle fibers to the center of the cell. We call it the equator of the cell. Looks like the Earth here, there's the equator. So we say the sister chromatids are aligned on the equatorial plate or metaphase plate. And these spindle fibers, a lot of these are attached right to the centromere. So far we've gone through prophase, we're in metaphase, and the next phase is anaphase, where we take our sister chromatids and separate them. So let's take a look at that. There we go. Sister chromatids are separating. When sister chromatids separate, each one of these now becomes a chromosome. It was one chromosome when the sister chromatids were attached. Then when they separate, each becomes its own chromosome, and they're exactly identical to the other sister chromatid. Well, these sister chromatids move to opposite poles during anaphase. Then you get to the last phase, which is telophase. And in telophase, your chromosomes here and chromosomes here are going to be surrounded by a nuclear envelope again. So there we go. The nuclear envelope reforms. The chromosomes go back to being chromatin, or loosely wound around the histone proteins. The cell then goes through cytokinesis, and you can see the cleavage furrow on each side, which is an indentation for cytokinesis to take place. The cell divides and you get two cells with the exact same genetic content by mitosis. Let's take a look at this sped up. Yeah. So now let's go ahead and go through the different phases. So prophase, we see that the chromatin are condensed to form visible, with a light microscope, chromosomes. Go from not being able to see the chromosomes here, chromatin, to condensing down in prophase to form chromosomes. Each of these chromosomes consists of identical strands called sister chromatids that are held together at a region, constricted region, known as the centromere. During prophase, the nucleolus and nuclear membrane, remember the nucleolus produces the ribosomes, this nucleolus and nuclear membrane are going to disappear. The centrosomes are going to move to opposite poles as they produce the spindle apparatus, also called the mitotic spindle, which are a network of microtubules for the movement of chromosomes during mitosis and consist of the following. We have kinetochore microtubules or kinetochore microtubules. These attach to the kinetochore, which is a molecular motor on each side of the centromere. So when you're looking at the centromere here, inside that centromere is a little molecular motor called a kinetochore. The kinetochore microtubules are going to attach to the kinetochore. And here's the kinetochore. It's a little tiny molecular motor that we find in the centromere. And basically what's going to happen is that this kinetochore, this is going to help move the sister chromatids along that spindle fiber. The second type of microtubules we have are polar microtubules. And these expand to opposite poles and lengthen the cell. Polar microtubules are oriented parallel to each other, but in opposing directions. So you have one polar microtubule coming this way, kind of like reaching for the other pole, and you have the other polar microtubule reaching this way. These are crucial for pushing the centrosome and spindle apparatus apart during cell division. They have little tiny motors right here, and so what they do is these little molecular motors are moving the microtubules in opposite directions. So they push the cell apart. Let me show you a video on how that works. And here we have a cell that's in metaphase. These are the chromosomes at the center. And these polar microtubules are going to help push the cell apart as it divides. Take a look at this. You see the cell moving apart? That's thanks in a large part to those polar microtubules. The other type of microtubules we have are the aster microtubules. These are short microtubules that are attached to the cell membrane, cytoskeleton, and they anchor the centrosome into position. They get their name aster for asteroid because they have these little streamers coming out. These are the microtubules, and they help to anchor that centrosome in place as it builds these other microtubules. And you have the movement of those chromosomes along the microtubules. The next phase is metaphase. In metaphase, those sister chromatids joined to the centromere are aligned on the metaphase plate, also called the equatorial plate. And here's a cell right here. Ooh, and you can even see these. Look at this. These are those aster microtubules. These are those other microtubules, kinetochore microtubules, those polar microtubules. You can't tell them apart in this. But then look at those sister chromatids aligned on the metaphase plate, also called the equatorial plate. So in metaphase, those spindle fibers align the chromosomes, each consisting of the sister chromatid, on the metaphase or equatorial plate. Anaphase. This is where the sister chromatids are going to separate at the centromere, forming two sets of identical chromosomes. Note that at the time of separation, each chromatid is now referred to as a chromosome. So as soon as these separate, these chromatids are now chromosomes. The daughter chromosomes move toward the opposite poles of the cell. Remember that kinetochore we were talking about? The kinetochore molecular motors walk, or they're going to pull those chromosomes along the attached microtubules, along with the microtubule disassembly at the kinetochore. So as a sister chromatid is moved over that kinetochore microtubule, the microtubule breaks apart into tubulin subunits. In late anaphase, we're going to see a cleavage furrow begins to form at the cell here, getting ready for cytokinesis. So the next phase is telophase. And in telophase, the nuclear membrane is going to form around each set of these chromosomes. The chromosomes are going to uncoil, becoming chromatin. The nucleoli reappear. and the cell prepares to divide. So you're going to get that continuation of the cleavage furrow, and you're going to get cytokinesis at the end of telophase. And cytokinesis is cell division. It divides the cytoplasm of the parent cell into the two daughter cells. It begins with the formation of this cleavage furrow during late anaphase and continues through telophase. The cleavage furrow is a constriction of the cell membrane along the center of the cell that eventually pinches the cell in two. You can see some cleavage furrow right here as well. It looks like a tightening of a belt around the waist. The end result? Two identical daughter cells, each with DNA that's identical to the DNA of the parent cell. That's mitosis. Mnemonic to help you remember mitosis: I've probably made all this correct. Ive is interphase, probably is prophase, made is metaphase, all anaphase, this telophase, and correct cytokinesis. Pause your video and see if you can go through and name these stages. Right here we have what phase? Prophase, because those chromatin have condensed down to chromosomes. This phase right here is metaphase. Our sister chromatids are aligned on the metaphase plate or equatorial plate. This phase right here is anaphase, because sister chromatids are separating. And this phase here, telophase. We have the nuclear membrane formed around each of these sets of chromosomes. You have the dividing of the cytoplasm, the cytokinesis. Any questions, let me know. I'll be happy to go over this with you. And you want to be able to explain mitosis to somebody. It helps when you explain things to somebody else. It really helps to solidify what your knowledge is. And if it's a time when you don't have somebody to talk to about this, well then talk to your cat, talk to your dog, talk to your children about it. Make it a bedtime story. Ooh, the adventures of mitosis, the adventures of cell division. You can even explain it to your plants. Oh my gosh, just talk about it. When your brain hears you talk about this, it's going to pick it up better, hearing your voice speaking about mitosis. So let's test our knowledge. Go ahead and pause the video and select the term that makes the sentence correct. Here are the answers. Go ahead and pause the video, select the term that makes the sentence correct. Here are the answers. Pause the video, select the term that makes the sentence correct. Here are the answers. Pause the video, select the term that makes the sentence correct. Here are the answers. Let's pause the video, select the term that makes the sentence correct. And here are the answers. Go ahead and pause the video, select the term that makes the sentence correct. Here are the answers. In the next video, we're going to be going over the control of the cell cycle. And if you have any questions at all, don't hesitate to let me know.